This is the University of Hawaii Maui College, the college on Maui.
So today we're going to talk about neurotransmission. Well, and I know you guys all remember this from your thorough understanding of anatomy and physiology, the neuron is the basic cell of the nervous system. And I'll tell you right now that when we get to exam 3 and do central nervous system pharmacology, you'll need to remember all of the information from this lecture today. So this lecture today is like 2 in 1. And the neuron is the basic cell of the nervous system. Neurons provide an instant method of cellular communication. And so here is my cartoon of a neuron. Sure, neurons come in all sorts of shapes and sizes, but this will be the neuron we use today. And there are unimaginable variations in neurons, variations in neuron structure and function, but there are many common features that neurons share. And here is the cell body of the neuron. There's the nucleus in the center. And the cell body of the neuron carries out exactly the same functions as what we talked about in cellular physiology. And coming from the cell body is an axon. The axon is a long tube that carries an electrical signal, and that electrical signal is called an action potential. That's why it shows that little lightning bolt going across the axon. That's supposed to show the electrical signal going down the long tube that's called an axon. Not too complicated. So an action potential is the electrical signal that is carried down the axon. Something I want you to keep in mind is the action potential is only carried one way down the axon. Please do not confuse the term nerve with neuron. A nerve is a collection of neuron axon fibers. And the signals in nerves can run in both directions. Efferent, meaning going out from the central nervous system. Afferent, sensory, coming in. And some nerves have efferents and afferents within the same nerve. But a neuron, its action potential only goes one way. And so if that action potential is headed away from the central nervous system, then it must be signaling either muscle or organs. If it's afferent coming into the central nervous system, it must be a sensory neuron. And so if you took a slice through a nerve, you would see the little neuron axon fibers in the center, but it wouldn't be lit up like a Christmas tree. All right, so here's my neuron, the cell body. The axon carries that electrical signal called an action potential. And that action potential, that electrical signal, causes neurotransmitter to be released from something called the synaptic terminals. We'll talk about that. And so these little red dots coming from my neurons, they're supposed to be neurotransmitter that comes from the neuron when it's being signaled with this action potential. So action potential causes neurotransmitter to be released from the synapse. So let's take a close view at where the neurotransmitter is released, these synaptic terminals. And here is the end of the neuron where the neurotransmitter is released. And here, the little red dots are supposed to be neurotransmitter, and they're packed in little packets called vesicles, and they're outlined in blue. So when the neuron fires, when the neuron has an action potential, goes down the axon to the end of the neuron, this action potential causes the vesicles to release neurotransmitter into the synapse. And so these little red dots are the neurotransmitter being released from the neuron. And the neuron can signal muscle. It can signal an organ like the heart or the lungs, or it can signal another neuron as well. And so this little space right here between the neuron and what it's signaling, that little space is called the synapse. So the neurotransmitter is released into the space called the synapse. And so you'll hear about presynaptic side of the membrane. That's this side where the neurotransmitter is released. The postsynaptic side of the membrane, that's where the neurotransmitter is signaling the muscle, the organ, or another neuron. So on the postsynaptic side of the membrane, down here, they look like little teeth. These are receptors, protein receptors, membrane-bound protein receptors, and these receptors are going to sense the neurotransmitter. So we can think of the neurotransmitter as being a key, and we can think of these little receptors as being the locks that these neurotransmitters will affect. So neurotransmitters, this should be in bright yellow. Neurotransmitters are chemicals released from neurons to communicate with either muscles, organs, or other neurons. What we're going to talk about mostly in autonomic pharmacology is neurons that communicate with organs and muscles. When we talk about the central nervous system, meaning your brain and your spinal cord, then we'll talk a lot about neurons that communicate with other neurons. Some neurotransmitters we're going to talk about in this course are these right here, acetylcholine or acetylcholine, depending on who you argue with. The catecholamines, dopamine, norepinephrine, and epinephrine, serotonin, and GABA, gamma-aminobutyric acid. These are the neurotransmitters that we're going to talk about in this course. We might throw some glutamate in there for those keeping score. But these are the neurotransmitters that we're going to talk about in this course. And so let's start with the neurotransmitter acetylcholine. And this is an actual email I received once. Dr. Farmer, I am tired of playing Where's Waldo with acetylcholine. And after I got that email, I thought, well, let's play Where's Waldo with acetylcholine. So where is the acetylcholine that we're going to talk about in this course? Well, one place, first of all, acetylcholine is made from acetyl coenzyme A. Acetyl coenzyme A is part of the mitochondrial energy transfer system. So when you go look at Krebs cycle and electron transport, you'll know all about acetyl coenzyme A. And there's a lot of multitasking in biologic systems. So we take this acetyl coenzyme A from the mitochondria and add that with choline to give us acetylcholine. Lecithin is a substance that has fatty acids that contain choline. That's why you'll see lecithin talked about improving memory because acetylcholine is a neurotransmitter used in the brain. So acetylcholine is made from acetyl coenzyme A and choline. So where are we going to find the acetylcholine in this course? Well, one of the places we're going to find it is the neuromuscular junction. Neuro, from the neuron to the muscle. So the neuromuscular junction is the place where the neuron signals voluntary muscle to move. And every voluntary muscle in your body is signaled by the neurotransmitter acetylcholine. And those receptors that sense the acetylcholine in the neuromuscular junction, we call those receptors nicotinic. And so here's my cartoon again of the neuromuscular junction. And so here is the neuron. We call it the lower motor neuron for those of you keeping score. And that lower motor neuron is what signals voluntary striated skeletal muscle. And the neurotransmitter is always acetylcholine, and the little receptors are always nicotinic receptors, and they're called nicotinic receptors because nicotine sticks to them so well. And just to make this point, there's a disease process called myasthenia gravis, and this is where antibodies are going to block the receptors at the neuromuscular junction. So when we talk about autonomic pharmacology, you're going to notice that there is some neuromuscular junction pharmacology involved as well. And so these little green Y shapes are antibodies. We'll talk about that in immunology, and they block the nicotinic receptors, block the acetylcholine from getting to the receptors, and this is why myasthenia gravis causes weakness. And I think I said this. Myasthenia gravis is weakness caused by circulating antibodies that block acetylcholine receptors at the postsynaptic neuromuscular junction, inhibiting the effect of the neurotransmitter acetylcholine. I liked the cartoon much better. Well, where else are we going to find acetylcholine? And this is what makes this all so complicated, because acetylcholine is used in the neuromuscular junction, but acetylcholine is also used by the autonomic nervous system. We're going to talk about autonomic nervous system next time, so be sure and bring your thinking cap with you. But we're going to talk a little bit about that today. And part of the autonomic nervous system that we'll talk about next time is the parasympathetic nervous system. So the neurotransmitter that signals parasympathetic smooth muscle movement is acetylcholine. So acetylcholine is what signals the heart to slow down. Acetylcholine is what signals lung airways to constrict. And those receptors are called muscarinic receptors because something called muscarine, it comes from mushrooms, sticks to those receptors so well. So here is my postganglionic parasympathetic neuron. We'll talk about those next time. And again, what's being released from that neuron? Acetylcholine. And this is how organs like the heart, the lung, the pupils are signaled by the parasympathetic nervous system. And the receptors are called muscarinic receptors. And we're going to talk more about that in the next lecture. We're also going to find acetylcholine in the central nervous system, and central nervous system is for exam 3, so we're not going to cover too much of the central nervous system in this section. However, many of the drugs that we're going to talk about in autonomic pharmacology are going to affect the central nervous system meaning brain and spinal cord, and so that's somewhere else you'll find acetylcholine. So where we're going to find acetylcholine, neuromuscular junction, that's important. Parasympathetic neurons signaling the organs, that's important. And the central nervous system as well. So that's Where's Waldo of acetylcholine. Let's talk about the catecholamines. I'm going to try to convince you, because there's a col right there and a col right there, I'm going to spend the entire exam trying to convince you that acetylcholine is a catecholamine. And those of you who take exams cold, trying to match up the col words, you're going to have a real difficult time with that. So do not be fooled. Acetylcholine is not a catecholamine. The catecholamines are dopamine, norepinephrine, and epinephrine. They are a family of neurotransmitters made from tyrosine. You need tyrosine to make dopamine, you need dopamine to make norepinephrine, and you need norepinephrine to make epinephrine. These are the catecholamines. And catecholamines are made from the amino acid tyrosine. I'll just switch over to the Elmo. There it is. And I wanted to show you right now, I renamed these 2.1 reading scans because we're covering them today instead of next time, but it's still the information. I want you to take a look at those and see how we've taken the amino acid tyrosine and turned it into DOPA, L-DOPA. That was the star of awakenings. We'll use L-DOPA as a drug in Parkinson's patients later. But notice we take that DOPA and make dopamine, and notice we take the dopamine and make norepinephrine, and we take the norepinephrine and make epinephrine. And so these are the catecholamines. If you look very carefully, you'll see the metabolism of tyramine as well. So I wanted you to take a look at these. You can memorize all the chemical steps if you want to. So the catecholamines are made from the amino acid tyrosine. Oh, and there's a picture of L-tyrosine. I showed you L-tyrosine a couple of times. We took phenylalanine, and through biotransformation, we oxidized it to make tyrosine. And tyrosine is what we use to make the catecholamines, and I can assure you that is something that you will definitely need to know, not only for my tests, but for board questions as well. So where are the catecholamines? Well, we're going to use them again in the autonomic nervous system, and we'll talk about the autonomic nervous system next time. But the other half of the autonomic nervous system is the sympathetic nervous system, and the sympathetic nervous system uses catecholamines to do things like speed heart rate up, open lung airways, and we'll talk about that next time. And the main neurotransmitters that we'll talk about in autonomic pharmacology are epinephrine, which is the same thing as adrenaline, and norepinephrine, something I call noradrenaline. And so if you can remember that epinephrine and norepinephrine are used by the sympathetic adrenergic nervous system, then you'll be doing great. And those receptors are called alpha and beta. And so later on, when we talk about high blood pressure and talk about beta blockers and alpha blockers, this is the reason right here, because we're trying to block the sympathetic nervous system. So where are you going to find the catecholamines? Autonomic nervous system, sympathetic neurons that signal the organs. Those sympathetic neurons are going to use epinephrine and norepinephrine, and the receptor types are alpha and beta. But I like cartoons better, so here is my postganglionic sympathetic neuron releasing norepinephrine into the synapse. And like with blood vessels, vascular smooth muscle, these are alpha receptors. And we can just take the same cartoon, and in the heart and the smooth muscle of the lung, norepinephrine is released from the sympathetic neuron, and beta receptors are what sense the catecholamine. And we also have dopamine and norepinephrine in the central nervous system. And again, we talk about the central nervous system when we go to exam 3. However, many of the things that we talk about in autonomic nervous system pharmacology affect the central nervous system. And so I wanted you guys to be on the lookout for that. So when we talk about autonomic nervous system pharmacology, the neurotransmitters we're going to talk about are acetylcholine and the catecholamines, and we're going to talk a little bit about dopamine, but mostly norepinephrine and epinephrine. So really the only 3 neurotransmitters we have to worry about for the next few weeks are acetylcholine, norepinephrine, and epinephrine. And please do not be fooled. Acetylcholine is not a catecholamine. Dopamine, norepinephrine, and epinephrine are catecholamines. We'll talk about serotonin and gamma-aminobutyric acid when we talk about central nervous system pharmacology for exam 3, and that's a few weeks away. Well, let's talk about breakdown enzymes and reuptake pumps. Well, we showed you how the neurotransmitter gets into the synapse, but if the neurotransmitter just stays in the synapse, then the signal just goes on and on and on. And we want the signal to stop as soon as the neurotransmitter quits being released. So those neurotransmitters have to be broken down, and broken down quickly. Think about fine muscle control. When acetylcholine is released to signal a muscle, then we want the muscle to move. And when the acetylcholine quits being released, then we want the muscle to stop immediately. And so the way to do that is to break down the acetylcholine in the synapse. And so we're going to talk about all sorts of reuptake pumps and breakdown enzymes going on in the synapse. So to get neurotransmitter out of the synapse, it either has to be pumped out or broken down. In some cases, it's just available for the extracellular fluid to just carry it away. And we can talk about that as well. So this is an old animation I did a long time ago. This is the neuron. That's the axon, and this is the synaptic terminal where neurotransmitter is going to be released into the synapse, and then this is the postsynaptic side where the receptors are going to sense the neurotransmitter. And so the axon is going to flash, showing that neurotransmitter is being released. The neurotransmitter is released into the synapse, comes in contact with the receptors, and signals the postsynaptic side of the membrane of the signal, whether that's a neuron or an organ. We'll notice that as long as the neurotransmitter is sitting in the synapse, then the signal just goes on and on and on and on and on, just like these lectures. And so how do we make it stop? Well, the neurotransmitter has to either be broken down or pumped out to get out of the synapse. Here we should have little pumps, little breakdown enzymes that magically appear, and you'll notice that they are pumping the neurotransmitter back out of the synapse to stop the signal that the neurotransmitter caused. So we can either break down the neurotransmitter or just pump it out of the synapse, and in some very rare cases the fluid can just carry it away. Well, what if we block all those little pumps? What if we block the enzymes that break down the neurotransmitter? Well, then the neurotransmitter is released into the synapse and continues to work longer. And so blocking the reuptake pumps, blocking the breakdown of neurotransmitter is going to be a strategy that we use in pharmacology, and we're going to talk more about that later. But I thought I would at least introduce you to this concept. I know I'll mention this a zillion times, and half of you will miss it on the test anyway, because this is a very difficult concept to understand. But acetylcholine, I told you what that was. That's the neurotransmitter that signals the neuromuscular junction, signals organs in the parasympathetic nervous system. But acetylcholine esterase breaks down acetylcholine. Acetylcholine esterase is what gets the acetylcholine out of the synapse. It makes the acetylcholine stop signaling the muscle. It makes the acetylcholine stop signaling the organ. So acetylcholine esterase is like a garbage disposal that breaks down the acetylcholine in the synapse. So if we block acetylcholine esterase, if we clog the garbage disposal that eats the acetylcholine, we will end up with more acetylcholine in the synapse. So if you know that things that inhibit acetylcholine esterase result in an increase in acetylcholine, then you're going to be doing better than 99% of people who ever take a pharmacology class. Later on, when we get to central nervous system pharmacology, we'll talk about antidepressants like Zoloft and Lexapro and Prozac, and those are SSRIs. Those are selective serotonin reuptake inhibitors. So I showed you the pumps that pump neurotransmitter out of the synapse, and we can block those pumps to leave more neurotransmitter in the synapse. And that's what we do with selective serotonin reuptake inhibitors, but we're not going to talk about these for several weeks. But I did want to introduce these concepts to you today. So let's talk more about the receptors. A certain type of receptor we'll talk about are gated ion channels. Opening an ion channel like a sodium channel, we're going to talk a lot about sodium channels in this course. Opening ion channels like sodium channels causes a voltage change across the membrane. And the bottom line with that voltage change is it gives us an instant on and off form of communication. And so a place we'd see gated ion channels is like in the neuromuscular junction. When the neuron fires to tell the muscle to move, we want it to move immediately. And when the neuron stops signaling the muscle, we want it to stop immediately. And so the way acetylcholine works on nicotinic receptors in the neuromuscular junction is to open one of these sodium channels. So opening sodium channels just causes an instant voltage change across the membrane, gives us an instant on and off form of communication. So here is the postsynaptic membrane, and here is the neurotransmitter in the synapse. And when the neurotransmitter comes in contact with the receptor, opens a sodium channel and allows sodium to rush into the cell, causing a voltage change across the membrane. And you'll hear these, if you read carefully enough complicated books, you'll hear it called ligand-gated ion channel. Channel meaning a hole in the membrane allowing an ion like sodium to rush into it. Gate means that the ligand, some chemical like a neurotransmitter, opens the gate to allow ions through the channel. And we're going to talk a lot about sodium channels and calcium channels when we talk about neurotransmission and when we talk about cardiac action potential as well. Well, not all neurotransmitters just open ligand-gated ion channels. Most of them do this. They activate something called a second messenger system. And so we're going to talk about second messenger activation. It's going to get real complicated. But what I wanted you to remember is that second messenger activation causes a gradual metabolic change. So something like speeding the heart rate up, something like signaling smooth muscle. That's not an instant on and off thing that we want. That's a gradual metabolic change. So you'll notice that drugs that affect smooth muscle, drugs that affect heart rate, they're going to activate second messenger systems. And so the second messenger system allows for gradual metabolic changes. So here's my favorite second messenger system. Here, a neurotransmitter, instead of opening a sodium channel and allowing sodium to rush in here, the neurotransmitter does something really complicated. When coming in contact with the receptor, it activates a G protein. You might read about that if you have Katzung's book. But the bottom line is here the neurotransmitter activates an enzyme called adenylyl cyclase, YL, adenylyl cyclase. And adenylyl cyclase converts ATP into a second messenger called cyclic AMP. ATP is that energy source that's made by the mitochondria. You guys know about that. And we can do all sorts of things with ATP. And one of the things that we can do with all that ATP is to make cyclic AMP. Cyclic AMP being the second messenger. And so activating adenylyl cyclase causes cyclic AMP levels to go up. And so you might read about a neurotransmitter increasing cyclic AMP levels. Or you might read a neurotransmitter doing exactly the opposite, stopping adenylyl cyclase and lowering cyclic AMP levels. And so there's all sorts of possibilities with that. But here our neurotransmitter has activated adenylyl cyclase, and the adenylyl cyclase is converting the ATP into more and more and more cyclic AMP. Well, the cyclic AMP can do all sorts of things. The cyclic AMP can go inside of the cell and activate all sorts of different regulatory proteins. And that can lead to receptor regulation, metabolic regulation, DNA regulation. The limits are endless and limited only to your imagination. Cyclic AMP does all sorts of things with all sorts of regulatory proteins. And not only does cyclic AMP activate many of those proteins, cyclic AMP will come along and deactivate all sorts of other regulatory proteins as well. So when we open an ion channel, it was real simple. We caused a voltage change across the membrane, and when we stopped doing that, the voltage change stopped. However, when we activate a second messenger system, we can cause all sorts of metabolic changes to occur. And for those of you completely lost, just try to remember that cyclic AMP is made from ATP, and cyclic AMP is a second messenger. Well, to make the story just go on and on and on, phosphodiesterase, there are all sorts of phospho and esterase words in pharmacology. Please do not confuse them with each other. I'm going to try to convince you on the test that organophosphates have something to do with phosphodiesterase. They don't. All right, phosphodiesterase, abbreviated PDE, it's what takes the cyclic AMP back out. It breaks it down. This is the garbage disposal inside of the cell that will lower cyclic AMP levels. And phosphodiesterase is inhibited by caffeine. And so here's my phosphodiesterase breaking down all that cyclic AMP. And then here are my 2 happy morning cups of coffee stopping the phosphodiesterase. And by blocking the garbage disposal that breaks down cyclic AMP, I can increase cyclic AMP levels in a different way. So when this second messenger story started, we increased cyclic AMP using a neurotransmitter that activated adenylyl cyclase. We can also increase cyclic AMP by blocking the thing that breaks the cyclic AMP down, phosphodiesterase. I know that's real complicated for a lot of you, but I just wanted you to be introduced to these concepts because they talk about them all the time in all sorts of nursing drug guides and healthcare drug guides, and they just assume that you know everything about phosphodiesterase and cyclic AMP. So I thought we would at least cover it a little bit. And to keep in mind, activating second messenger systems causes all sorts of metabolic changes. They're going to be more gradual and long-lasting than the instant on and off ion channels. And there are more second messenger systems out there than just the cyclic AMP. However, cyclic AMP is the one that you will see the most of, and cyclic AMP is the one that I have the cartoon of. And maybe we'll have more second messenger systems for me to cartoon one day. You might hear these receptors referred to as ionotropic, meaning it opens an ion channel, and we showed you that opening the sodium channel. Or your receptors can be metabotropic, meaning they activate a second messenger system to cause some kind of metabolic change. So keep in mind some of the drugs that we're going to talk about are going to affect ion channels. Other drugs that we talk about in the central nervous system are going to activate or stop second messenger systems. It's just this simple right there. Well, let's talk about action potential. Sodium, potassium, chloride, and calcium, these are the major ions of action potential. And they're each going to have their own ion channels when we go through this. Sodium chloride, boy, we've talked about that before. We'll talk about it again. It's table salt. You can find it at the grocery store. You can find it in the ocean. And there's plenty of it right there in your blood as well. We put the sodium chloride into water. We end up with sodium ions and chloride ions. And we're going to talk about sodium ions and chloride ions when we talk about central nervous system pharmacology and autonomic pharmacology all the time. And so sodium chloride, table salt. Sodium ions, chloride ions, the salt in the water. Potassium chloride is a salt as well, despite what they put on the can at the grocery store. Potassium chloride is a salt. When we put the potassium chloride into water, we get potassium ions and chloride ions as well. So sodium and potassium are very important ions in neurotransmission and action potential, and I think we're getting ready to talk about that. Well, I've talked about this before. We'll talk about it again. The sodium-potassium pumps. Sodium-potassium pumps are very important in membrane physiology, especially excitable membranes. And so these are the sodium-potassium pumps. Sodium-potassium pumps pump sodium out of the cell, and they also pump potassium into the cell, and they use ATP as an energy source. That's why you'll hear them called sodium-potassium ATPase, the sodium-potassium pumps. Here's my cartoon of a sodium-potassium pump, sodium-potassium ATPase, and here is my cell membrane. And you will notice the sodium-potassium pump is pumping sodium out of the cell and pumping potassium into the cell. So if you think about it, for those of you in the healthcare business, we consider the serum to be an extracellular fluid. We consider the serum to be outside of the cell. And if you look at a serum chemistry, you'll notice the serum sodium somewhere around 135, and you'll notice the serum potassium is somewhere between 2.5 and 3. And the reason there's lots of sodium outside of the cell and not much potassium outside of the cell in the extracellular fluid is because of the sodium-potassium pumps. And so when you take a look at a serum chemistry, you can see those sodium-potassium pumps in action. All right, so here's my sodium-potassium pump, pumping sodium out of the cell, pumping potassium into the cell. Don't forget, it's powered by the ubiquitous power of ATP. So what happens when we pump sodium out of the cell and pump potassium into the cell? Well, we end up with this sodium force. Where did it go? There it is. We end up with this sodium force where sodium is trying to get back inside of the membrane. And so the sodium-potassium pumps create a sodium workforce. It's a sodium gradient. And so how the sodium-potassium pumps do this is to form a gradient. By having more sodium outside of the cell, the sodium tries to get back in with a concentration gradient, and it tries to get back inside with an electrostatic gradient as well. Let's see where that's. And so something we can do with that sodium force is put that sodium to work. And so we can have all sorts of little sodium channels to allow the sodium back into the cell, and we can put that to work. Is that the slide I'm looking for? No. There it is. For those of you keeping score, the sodium-potassium pumps turn the cell membrane into a capacitor that stores electrical energy, and the entire membrane can use that electrical energy to do work. So for those of you who've had physics, know what a capacitor is. The sodium-potassium pump turns the membrane into a capacitor. For those of you who have no idea what a capacitor is, you can think of the sodium-potassium pump as creating a battery across the membrane, a source of energy that can be used across the membrane. And so we can use this electrical energy to do work. It's like that sodium force, that sodium trying to rush back into the cell because of its concentration gradient and the electrostatic gradient because it's positive outside of the cell. It's pushing the sodium back in because there's more sodium outside of the cell. The sodium's trying to rush back into the cell. And we can use that sodium force to do work, like transport nutrients across the membrane. And so here we're using sodium to transport glucose into the cell. And so it takes energy, it takes work to take a large molecule like glucose and get it into the cell. And the energy source to pull that glucose into the cell is that sodium force. And that sodium force was created by the sodium-potassium pumps. Well, the amino acid tyrosine that we're going to use to make the catecholamines. That's an amino acid that has to be transported into the cell using the sodium force. So that sodium force provides the energy to get things like amino acids, especially tyrosine, into the cell. We can use that electrical energy to do work like powering action potential. That electrical signal that goes down the axon, that's powered by the sodium force that's created by the sodium-potassium pumps. And I've shown you this before. When the neurotransmitter comes in contact with its receptor, it opens a channel, a sodium channel, and allows sodium to come in. By moving the positive charge across the membrane, we change the voltage across the membrane, and that is how an action potential occurs. So an action potential is a wave of voltage change that occurs by opening and closing these ion channels. And we can characterize that wave as sodium rushing in, and then potassium compensates by rushing back out of the cell. And so as we go through all this, if it gets too complicated, if you can remember this slide right here, an action potential is a wave of voltage change that occurs by opening and closing the ion channels. Action potential is just sodium ions rushing in and the potassium ions rushing out to repolarize, to reset the membrane. So let's see if we can get through this cartoon right here. Here's my sodium-potassium pump. The sodium-potassium pump goes all the time, all right? Unless we poison it, the sodium-potassium pump just runs continuously, creating that membrane battery, making the sodium ions on the outside, the potassium ions on the inside. It's positive on the outside, more negative on the inside. So here, our sodium-potassium pump is doing its job to create that sodium force. And here comes some agonist, some kind of neurotransmitter, and when it comes in contact with its receptor, it's going to open a sodium channel, in this case, and allow sodium to begin to rush into the cell. Well, when sodium begins to come into the cell, that changes the voltage across the membrane, because it used to be more positive out here, and now moving that positive charge across the membrane changes the voltage. Well, changing the voltage across the membrane is going to activate this little sodium channel right here. Notice that little lightning bolt means it senses a voltage change. So when we talk about gated sodium channels, some of them are going to be opened by neurotransmitter, ligand-gated ion channels, ligand-gated sodium channels. That means sometimes a chemical opens them up. But other sodium channels and other ion channels we'll talk about, they're going to be opened because they sense a voltage change. So if you read about this stuff carefully, you'll read about gated ion channels. Some of those are ligand gated, meaning they're opened with neurotransmitter. Others are voltage gated, meaning that they open in response to a voltage change. So here my agonist opened the sodium channel. The sodium began to rush in, causing a voltage change. And over here is a sodium channel that's gated by voltage, a voltage-gated sodium channel. And it senses that voltage change, and it opens and allows a big rush of sodium into the cell. Sometimes you'll hear these called fast-acting sodium channels. Well, with all the sodium rushing into the cell, that completely takes away our capacitor. It completely depolarizes the membrane. It started polar, meaning one set of charges were on the top and the negative charges were on the bottom. After we've let all this sodium in, there's a voltage difference across the membrane. And when that happens, something happens over here. Our potassium channel, notice the little lightning bolt on the potassium channel. That means it senses that there's been a voltage change across the membrane. Now that we've let all the sodium in, now that we've let a lot of sodium in, there's not the voltage change across the membrane, so these channels quit letting sodium in because the sodium doesn't need to rush in anymore. It already has. However, now the potassium channel senses that there has been a large voltage change across the membrane, and our potassium channels are going to open by voltage. So you'll read about gated potassium channels, and that's what we're talking about right now. And what that potassium channel does when it senses that voltage change is open up, and the potassium begins to rush out because of the concentration gradient of potassium on the inside. So because all that potassium in the inside, the potassium begins to rush out, and those positive charges leaving the cell is is what resets the membrane so that can all happen again. I know this is real complicated for a lot of you, but pharmacology talks about these things. If you can get a small handle on any of this, I think you'll have a lot better understanding of what we're going to talk about in the next couple of months. And so here's my cartoon of it. This is an action potential going down the axon of our neuron. And so when it's blue, that's sodium channels opening, and the red is the potassium rushing back out, resetting the membrane so that if the action potential continues, sodium can rush in, potassium channels opening allow the potassium out to reset, and then sodium rushing in, potassium rushing out, sodium rushing in, potassium rushing out. And it's that wave of voltage change that occurs that causes the action potential. In your notes, if you looked at them more than a few hours ago, they were called 2.2 reading scans, but I wanted to cover them today, so I renamed them 2.1 reading scans, and I wanted you to take a look at them because we see these drawings in all sorts of pharmacology books, and they just assume that you know everything about what they're trying to talk about. And so here we're going to look at the acetylcholine synapse, and this is in your resources section. Right now it's called 2.1 reading scans. And so you should see a picture that looks like this, and this is our acetylcholine synapse. And so this is the neuron. I had a little picture of neurotransmitter being released from the neurons, and so this is the presynaptic side, this is the postsynaptic side, and this is the synapse here in the center. So let's take that picture and take this little part of the picture right here and blow that up and see what we get. Notice that to make acetylcholine, we have to get choline into the cell. And remember, we use that sodium force to pump choline into the cell. And so this is the point they're trying to make right here. That choline, to make acetylcholine in the neuron, the first thing we have to do is pump choline into the neuron, and we use the sodium force to do that. And notice hemicholinums block that. So this is a type of toxin that blocks choline from getting into the cell. Now, I'm not going to ask you anything on your test about hemicholinums, but what I did want you to see is already we have a way of blocking acetylcholine from coming from a neuron just by blocking choline from coming into the cell. And so that's the point that I wanted you to see right there. Choline being pumped into the cell with that sodium force, and already we have a type of drug, a toxin, that can block the choline from getting into a neuron that makes acetylcholine. And I showed you this picture of choline being transported into the cell by sodium. So now we've gotten this, the choline into the cell. We'll take a look at that part of the picture right there. Remember, we need choline, and that comes from things like lecithin. And then acetylcoenzyme A comes from our mitochondria, and those are put together to make acetylcholine. And so that's what I wanted you to see with this part of that picture. Well, now that we have acetylcholine, we need to pump the acetylcholine into the little vesicles, the little packets that hold the neurotransmitter. And if we take a step back, we can see Vasimicol blocks putting acetylcholine into the vesicles. So here again is another strategy to prevent the neuron from releasing acetylcholine by blocking putting acetylcholine into the vesicle. So we'll take a look at this right here. I wanted you to see this right here. Now that we have the acetylcholine in the vesicles, the vesicles wait for neurotransmission, and when action potential comes down, the vesicle releases the neurotransmitter into the synapse. And there's all sorts of things you can read about, these little snap proteins that make that happen. But what's very important is this right here. Notice calcium is required for vesicles to be released from the neuron. And so we need calcium for any kind of vesicle to be released from the cell. And this is something that I wanted you to keep in mind. Notice botulinum toxin also prevents acetylcholine from being released from the cell by blocking the release of acetylcholine. And I said this opening sodium channels allows for neurotransmitter release. This is important for you to remember. Botulinum toxin, we know about this from botulism. It's a rare but serious paralytic illness caused by a toxin. That makes sense. We use acetylcholine to drive our respiratory muscles. Our voluntary respiratory muscles are controlled by acetylcholine, and botulinum toxin will prevent the acetylcholine from being released. And so when we suffer from botulism, it causes paralysis and causes us to not be able to breathe. And it is produced by a bacterium, Clostridium botulinum. Botulinum toxin is one of the most powerful known toxins. About 1 microgram is lethal to humans, and it acts by blocking nerve function, by preventing the release of acetylcholine from the vesicles. And it can lead to paralysis and respiratory paralysis and death. However, we'll talk all about botulinum toxin when we talk about Botox injections to relax facial muscles and make us look younger. Well, let's take a look at this part of the picture right here, the synapse. And so here is my acetylcholine being released into the synapse after action potential occurs. And notice it comes and activates cholinereceptors. Acetylcholine receptors are also called cholinereceptors here on the postsynaptic membrane, the postsynaptic cell. And so the acetylcholine is going to activate these cholinereceptors. If this was a muscle, these would be nicotinic receptors. If this was a parasympathetic controlled organ, these would be muscarinic receptors. And notice these little pieces of broccoli on the picture, acetylcholinesterase. This is what breaks down the acetylcholine, turning it back into choline and acetate. So acetylcholinesterase breaks down acetylcholine. That's how we get the acetylcholine out of the synapse. I think this is on page 79 of one of the older Cat Sung books. I'm sure it's in one of the new Cat Sung books, and it's probably 10 times as complicated. So we'll take a look at that again. There we go. So my acetylcholine is broken down by acetylcholinesterase. The acetylcholinesterase breaks the acetylcholine down out of the synapse, turning the acetylcholine into choline and acetate. If we block acetylcholinesterase by using acetylcholinesterase inhibitors, we end up with more and more and more acetylcholine in the synapse. And this is what I want you to be able to see from that picture. And so you'll hear about cholinomimetic drugs, drugs that mimic acetylcholine. Nicotine and muscarine obviously signal acetylcholine receptors. Nicotine signals nicotinic receptors. Muscarine signals muscarinic receptors. And we're going to talk about all sorts of muscarinic and anti-muscarinic drugs when I open up the forum section day after tomorrow. But something else that mimics increased acetylcholine levels is blocking the garbage disposal, acetylcholinesterase inhibitors. And so I know this is real complicated for a lot of you, but this is what pharmacology is about, and it is certainly not my fault that we are such complex creatures it is your fault. Well, so now let's look at the catecholamine synapse. It says catecholamine back there somewhere. And the thing that I wanted you to see about the catecholamine synapse is that it really functions pretty much the same way, and the toxins function pretty much the same way. Now, catecholamines are dopamine, norepinephrine, and epinephrine. Catecholamines are not acetylcholine, despite what I try to convince you of on the exam. So let's look at the catecholamine neuron and see what's going on. Notice we start the same way. Here we're using the sodium force to transport tyrosine. An amino acid tyrosine is what we use to make the catecholamine. So here's the sodium force pulling tyrosine into the cell. And if we take a look at that part of the picture right there, we can see that we need, once the tyrosine is in the cell, we turn that into dopa and dopamine. And here, met-tyrosine blocks the conversion of tyrosine into dopa. I don't need you to know this. I just wanted you to see that already we have a way of preventing this neuron from functioning by preventing the amino acid to be converted into dopa. And we'll take a look at that picture right there. Here, we're taking dopamine and putting it into the vesicle, and we can take the catecholamines, norepinephrine, and put it into the vesicle. And here, we see reserpine blocks putting catecholamines into the vesicles so they can be released. Reserpine still is, but was widely used in the old days as a blood pressure medication. Catecholamines are part of your sympathetic nervous system, your adrenaline system, that make heart rate and blood pressure go up. And if we can prevent catecholamines from even getting into the vesicles, then we can lower blood pressure and heart rate. And reserpine prevents the catecholamine from getting into the vesicle, and so that's why I wanted you to see this picture right here. Again, we have another strategy to decrease nerve function. We'll take a look at that part of the slide right there. Notice again, calcium channels are required to allow the vesicles to release neurotransmitter into the cell. So we'll take a look at this part of the picture. Here we have norepinephrine being released into the synapse. And here we'll assume that it's some kind of organ like the heart or the lungs. Notice our norepinephrine comes in here and signals the adrenoreceptors. We'll call these alpha receptors and beta receptors. And notice the norepinephrine in this case, in some cases, especially in the sympathetic nervous system with heart and other organs, the norepinephrine can just float away. And the reason the synapse is open, allowing the norepinephrine to float away, is because when your adrenaline rush kicks in and you're all full of adrenaline, the epinephrine can come in and activate all these receptors and kick all that sympathetic business into overdrive. And that's what we're going to talk about next time. But again, notice drugs like cocaine, drugs like antidepressants, tricyclic antidepressants, are going to prevent norepinephrine from being taken back into the cell. They're going to prevent norepinephrine from being pumped back into the cell. And so these are going to be things that we need to keep in mind, even though we talk a lot about autonomic pharmacology, how these drugs affect the organs. We also need to keep in mind that they affect the brain as well in the central nervous system. Well, before we run away, we'll add one more thing on the Elmo. I showed you these two pictures right here. These are in your reading scans. I want you to take a look at those very carefully and make sure you understand them. I showed you how we take tyrosine and make dopamine dopa, dopamine, norepinephrine, and epinephrine. One thing I wanted you guys to also take a look at in your 2.1 reading scans is this right here. Let's see if we can unzoom that. There we go. take a look at this page right here. Notice that I can barely operate this thing. There we go. When you take a look at this page, I want you to see that here are the catecholamines epinephrine, norepinephrine, and dopamine. And something that I want you to see is what breaks down the catecholamines. How are the catecholamines broken down? That's what's on this page. And notice MAO, monoamine oxidase, breaks down the catecholamines, and we'll talk about that a lot during central nervous system pharmacology. But no matter what step you take in the breakdown of catecholamines, we're either going to have monoamine oxidase breaking down the catecholamines, or we're going to have COMT, catecholmethyltransferase. But the star of the show is monoamine oxidase, is what breaks down the catecholamine products, and our final products are homovanillic acid, abbreviated HVA, and you'll see that especially on a test question. And then 3-methoxy-4-hydroxymandelic acid, we call that VMA. VMA in here is not the Video Music Awards. VMA is a breakdown product of the catecholamines. So when things like epinephrine are released into the body from the adrenal, when that epinephrine is broken down, it's broken down into VMA and HVA. I wanted you guys to take a careful look at that on your scans. Well, that is more than enough for one day. When I come back, we're going to talk about 2.2 autonomic nervous system pharmacology. So until then, aloha.
This is the University of Hawaii Maui College, the college on Maui.